Hello, everyone. Welcome back to our Clinician Engineer Hub webinar series. Today we have with us Dr. Liana Shvachiy, and she will talk about the mechanobiology of bone. She's the professor and director of the Cellular Biomechanics Laboratory at University of Toronto, and with across appointments in the Department of Mechanistics and Industrial Engineering and the Institute of Biomaterial and Biomedical Engineering. Dr. Yu's major expertise is in the area of cellular biomechanics, bone cell mechanics, bone cell mechanobiology, bone adaptation, and bone biology. She has published over 70 peer-reviewed papers in very renowned journals, and also as conference abstracts in leading journals across multiple disciplines to date in the field. She is the recipient of numerous honors and awards, such as the Early Researchers Award of Ontario, Young Investigator Award of International Bone Fluid Flow Workshop, and the Leaders of Innovation Fund Award from Canada Foundation of Innovation. Thank you very much, Dr. Liana, to join us, and the stage is yours.
Okay, thank you for the introduction. Let me start to share my screen.
Yes.
Okay. Okay, can everybody see it? Okay, so today I will be talking about bone mechanobiology, but specifically focus on the microfluid platform we've been developing for the study of bone mechanobiology. So first, I would like to mention the personnel who contributed to the work that will be presented today. So this is my lab. So Christina May, she is the major researcher driven the study I will be presenting today, which is the microfluid platform for bone mechano sensitivity study. And also Dr. Kevin Middleton, he graduated 2 years ago. He is the person developed and also fabricated platform. So now, first I want to talk a little bit about the background, which is the loading effect on bone. Now, bone is a very, it's a solid structure. Most people think about bone like a supporting structure without realizing that the bone actually is a very dynamic structure. Exercise can have impact on muscles, in general on your health, but most people don't realize it also actually has major impact on the bone structure. As you can see here, in the playing arm of a tennis player, the bone is much denser and larger compared to the bone in the non-playing arm, indicating that mechanical loading can stimulate bone formation. On the other end of the spectrum, if we're looking at astronauts going through space travel, right, we will see that you will see that the astronauts actually lose bone a lot because of the microgravity environment, because lacking the mechanical stimuli during the entire space travel. So we've been wondering, we've been trying to understand how mechanical loading can affect bone structure and how that changed the bone structure. And there's many questions we've been investigated in the past 20 to 30 years. But recently we've been mostly focused on the last two questions, which will be the focus on today's talk. We ask, how does mechanical loading affect intercell population crosstalk in the regulation of other diseases such as diabetes, such as bone cancer metastasis? And also, we're trying to see whether we can develop a better in vitro platform to study bone cell mechanotransduction. So let's first look at what cells are living in the bone, right? So there's mainly three types of cells in the bone. There's osteocytes, which are the major cell population. About 90 to 95% of all bone cells are osteocytes in adult bone. And there's osteoblasts responsible for bone formation. osteoclast responsible for bone resorption. Of course, in the marrow, bone marrow, there's also bone marrow stromal cells, which can differentiate into osteoblasts given right chemical or physical conditions. Now, osteocyte has been accepted as the mechanosensor cells. These cells, they're very sensitive to mechanical loading. So I'm going to play two short video clips. On the left-hand side, you will see, you're looking at here is osteocyte seated on the glass slides, experiencing fluid flow, but this fluid flow is actually steady flow, unidirection. And this is actually 8 hours video compressed to less than 1 minute. And you can see that the cells are actually stretching a little bit, trying to move around a little bit, but rather relaxed. But if you're looking at the same cells sitting on a glass slice and experiencing dynamic flow, in this case is bidirectional oscillatory fluid flow at a frequency of 1 hertz. So even we keep the shear stress at exactly the same level, you can see that the cell morphology change a lot, cell mobility change a lot, cells are trying to reach out to each other. It's almost like they're trying to establish connections and form this community. And this is really a stunning finding for us. So we realized that mechanical stimuli is just critical for this type of cells. And they are very sensitive to mechanical loading, not only to the magnitude, but also to the type of mechanical loading. In this case, is the frequency, right? Okay, so one of the early signals that the cells can, bone cells can respond to mechanical loading, the second messenger we're looking at here is the intracellular calcium immobilization. So once you apply loading, could be shear stress, could be pressure, right? And you will see that instantaneously, the intracellular calcium concentration goes up, which is indicated as a pink color. So it's almost like a firework. So again, demonstrate that bone cells, osteocyte, responds to mechanical loading very quickly. And this is actually a real-time video. So next, so we look at we try to understand how mechanical loading can affect the bone cells. So the approach we use to answer or test the question, is using in vitro platform. So now the current in vitro flow system, typically we use parallel plate flow chambers, but it's just a chamber that can hold a glass slice where you can put the cells in and then apply fluid flow to the cells. Initially, this setup is this device is established for study endothelial cells mechanotransduction. Problem with that is that it lacks direct real-time communication between different cell population. Also, it lacks half-life signals interactions between different cell population. So, and then there's a co-culture study, all right, and using the transwell, trying to kind of like overcome some of the limits with the traditional condition medium based study. So with the transwell, of course, you can have the cell communicate in a real-time manner, but the problem is that it also lacks the capability of real-time imaging of the cell response, and you cannot really apply well-controlled fluid flow. So then there's a microfluid co-culture device being established in many other fields. The problem with that is with those devices available is that usually this flow device is the flow is isolated device have only one cell type being embedded in the gels. They're not necessarily physiologically relevant, and it only allow very weak signal gradients, which is actually quite critical for bone cell apoptotic function. So therefore, we decided to establish our own microfluid platform. So what's shown here, maybe we can just focus on this part. So basically, we developed this 3-channel microfluid device. The scale is indicated here. So basically, it's about 1 millimeter in diameter, but The depth is about 50 to 200 micrometer. And we can actually have different cell populations seated in these different side channels, but that between different channels, we actually have even finer side channels allowing the signal to get across. So that way we can actually apply different mechanical loading to different cell population at the same time, observe how they communicate to each other, and also different cell population are under different chemical signals with different chemical signal gradient. So we use the fluorescent beads to demonstrate that the signal can get across from this very fine side channels from one channel to another. We can establish the signal gradients beautifully, and that allow us to look at how cells communicate when there's a real-time communication between different cell population. And we have shown that if you put osteocyte without loading in one channel and osteocyte with loading on another channel, so this will be the loading channel, this is non-loading channel. We can put the osteoclast here, and we can look at how osteoclast, again, in this case is the bone resorption cell, can respond to different signals received or released by cells under different mechanical environment. And we show that osteoclast actually prone to move towards the non-loading side, which means that when bone cells is not loaded, they will release signals that will attract osteoclast and making more osteoclast formation, so that they will the bone will be more degraded, and then therefore you will lose more bone as a result. All right, and we're also trying to develop high throughput microfluid platform, so we can have this multiple co-culture microfluid platform with different channels, loading channels with different mechanical loading. In this case, it's 0.5 Pascal, 1 Pascal, 2 Pascal. So we can look at how cell response in different channels with different mechanical stimuli. And we can actually differentiate it very nicely. So for example, in the 2 Pascal channel, there's we first validated the velocity profile, and then we also look at calcium response. We found that in the 2 Pascal channel versus 1 Pascal, we have much higher osteocyte response with intracellular calcium concentration increase. So that is all set up the foundations of our work for the bone metastasis study, which is the focus of today's talk. So why we're interested in breast cancer and bone metastasis? Well, we know that breast cancer, bone metastasis is spread of cancers to bone. So 85% of advanced breast cancer patients has suffered from that. And there's a severe symptoms as a result. And the current treatment involves surgery, chemotherapy, radiotherapy. It can only relieve breast cancer bone metastasis symptoms, but can't really treat it. And also we don't have much options on how to prevent bone metastasis. So we know that mechanical loading is a very strong mediator or regulator of bone structure, and also osteoclast function is very tightly regulated by osteocyte. On the other hand, we also know that there's a crosstalk between bone cells, especially osteoclast and also osteoblast, to cancer cells. So we think it might be, right, when you have a mechanical loading, in this case exercise, you actually can have a positive impact in terms of preventing bone metastasis. So this is what we propose here in terms of hypothesis. So we have bone cells living in the bone matrix. When we have a mechanical loading, then because bone is a power structure, the interstitial fluid flow is being driven, then that will result shear stress applied on the bone cells that will leading to the regulation of releasing certain cytokines affecting both osteoclasts and also endothelial cells in the surrounding vasculature. That will in the end have a crosstalk with cancer cell, determine whether they will have extravasation, which is the process that the cancer cell escape from the vascular system, and then they migrate towards bone and then start this secondary tumor growth, which is bone metastasis. So that's the idea. Now, going back, so exercise has been recommended as a positive after like a post-surgery procedure for the patients. So the physicians usually recommend exercise to patients for the patient to do exercise to regain their physical activity, to regain their muscle strength. But limited studies show, or we don't really know what is the impact of exercise on bone metastasis. That's another question we're trying to understand here. So again, there's a 4 key player here. One is the osteocyte, which are the cell I just showed you in the video, which are very sensitive to mechanical loading. There's the osteoclast, which is the bone resorption cell. There's the osteoblast, which is the bone formation cell. And there's the endothelial cells. So endothelial is very important. If they become very sticky to cancer cell, there's a more chance your cancer cell will attach to the wall of the vasculature and then start extravasation. So we did a whole bunch of conventional condition medium study. We have demonstrated, given the limited time I have today, so I won't go to the details. I'll just give you the summary of what we learned from those studies. So we show that mechanical loading will affect osteocyte, which in the end will directly affect the cancer cell. But without any cell in between, if we're just looking at a direct crosstalk between osteocyte and breast cancer cells, we actually have the opposite of what we expect to see, which is the increase of migration and decrease of cancer cell apoptosis. So it's a big surprise to us, and then we realize something is missing in the picture. So what's missing here is some cells living kind of like in between, right? So between the osteocyte and cancer cells. In this case, if we put osteoclast here, we know that osteocyte can downregulate osteoclast differentiation when osteocyte experiencing mechanical loading, and that will result downregulation of cancer cell migration and promote cancer cell suicide, which is apoptosis. And again, if we have endothelial cells living in between and we do the double conditioning study, we show that mechanically challenged osteocyte can promote endothelial regulated downregulation of transendothelial migration and increase cancer cells apoptosis. So we also saw that osteocyte and mechanical loading can actually decrease the permeability of endothelium and also decrease the adhesion of the endothelium by breast cancer, and also in the end actually can affect the metastatic potential of cancer cells. So this actually through the signal released by the endothelial cells to regulate the cancer cell metastatic potential, in the end leading to the decrease of invasion. So all this is good, but all this is done in a conventional double conditioning setup. And in this setup, the problem is that we don't have the cells really crosstalk to each other in a real-time manner, which is what happened in the in vivo situation. Therefore, we developed our microfluid platform. So I'll just go to the details of the device we developed. So this is the microfluid device chip. We have this basically 6 co-culture microfluid channel system established. And then if you focus on one of the co-culture microfluid platform we have, this is a 3D view. If you take a closer look, so this is the schematic image showing what is the cross-sectional view of the system. So we basically have 2 channels. One channel, we can see the osteocyte there and apply mechanical loading. The other channel, we actually have, we can actually see the endothelial cell form this artificial vessel-like structure, and we can actually inject cancer cells there. And in between, there's hydrogel side field side channels, so we can actually have we can observe whether the cancer cell under certain signals received from the outside under mechanical loading, whether they can respond with migration towards through the side channel towards the outside channel, whether they can escape and cross endothelium formed here. So that's the idea. And we show that indeed, if we simply just put some chemicals that can attract the cancer cells, we can actually show that the fluorescent dyed cancer cells can actually migrate through the hydrogel, escape the endothelium here, across the endothelium, and then try to get across to move to the bone environment. So this is the setup. We have this custom-made pump allow us to deliver a well-controlled free flow to our microfluid system. And so this is the image showing we can form a very nice endothelium, like a vessel-like structure, artificial vessel. And we can show that if you look at the right fluorescent stain is a HUVEC, which is endothelial cells here, and we have green stained cancer cells. When they overlap, we will see the yellow color shows that the cancer cells are trying to escape, right? Get across the endothelium, and then we can see that also cancer cells actually escape and then enter the hydrogel field set channels. So we found that if we add flow to the outside channel, which will be on the left-hand side here, and we can show that it actually can dramatically decrease activation distance and activation percentage as shown here. This is the outside under mechanical loading. This is outside without mechanical loading. You see this dramatic difference. So again, so one may wonder whether what we have observed here is actually not caused by any signals released by outside, but actually simply just caused by the mechanical loading application. So in that case, we did another control study. Without having outside sitting in the outside channel at all, it's an empty channel, but we simply apply flow versus static. And we're trying to see whether that will make a difference. And we measure the distance the cancer cell travels, and also the percentage, how many channels will be occupied by the cancer cells. And for both, we don't see any difference. So between the static group and the flow group. So in that, we can conclude that what we observe in terms of difference between flow group and non-flow group in extravasation distance and extravasation percentage is indeed caused by outside under mechanical loading released cytokines or signaling molecules. So in summary, we established this very novel microfluid platform. We can actually expose bone cells to mechanical loading, and then we see that we'll directly real-time crosstalk with cancer cells, inject in the artificially made vessels in our system, and then we can show that activation distance and percentage is being downregulated. Yeah, so this work has been published in 2019. So it's actually also featured in the Scientific Translational Medicine. And this is actually the first platform allowing us to, in the field. So then with this platform, we're allowed to take a closer look on what's really going on for trying to answer the question, what happened when you have bone metastasis, and whether the loading can actually have any positive impact on preventing the bone metastasis. I think I'm running out of time, so I'll just end it here. And if you have any questions, I will be happy to answer the questions.
Thank you very much, Dr. Layden. If you want, you can continue because it's a very interesting talk, and we still have time.
Oh, okay. I thought that we have only 30 minutes.
No,
okay.
It's fine. It's fine.
Okay, so well, another thing I want to mention is another interesting setup, kind of like gadget we developed in our lab. So As we know that in the bone cell, in the bone tissue, when you have a loading, you don't generate uniform mechanical stimuli. And bone is a curved structure. When you apply loading, when you do the exercise, jumping, or any type of mechanical loading you apply to your bone, it's not going to generate a homogeneous field of force in your bone. So there's always a gradient in terms of force. So we decide to look at whether that will have any impact on bone cell behavior. So what we did was actually we have this local stimulation of ultrasonic setup. So this is actually so this is schematic video showing what we're trying to do here. So we can have this macro beam, right? So it's a magnetic beam. So we can actually using the magnetic field to control its swing. So when it swing back and forth, left and right, that will generate the fluid flow, right? So that will generate fluid flow shear stress. And we can have also side seated at a substrate, on the substrate. And then with adding this magnetic field manipulated beam swinging, we can actually very nicely control the force or the shear stress gradient. So we can see how cells will respond with high shear stress versus low shear stress. How do they communicate to each other? This is just the side view. So we have the magnetic actuator and go back and forth. And then this is a glass slice where the cells can sit on it. So, so this is the whole setup. So basically we have this magnetic field generator. We can put that around the microscope platform. And then this is connected to a computer system that allow us to generate well-controlled magnetic field and try to control the magnitude or the frequency of the magnetic beam swing. So what we can see here is that we can actually mapping the magnetic beam swing magnitude and also the shear stress level. So the shear stress level is also indicated by the color. So if you look at the red color, that means you actually have quite high shear stress. Sorry, this is response time. The shear stress level, the higher, the lighter, the yellower, it will show it's actually higher shear stress. So with this, you can see that one is close to the high level shear stress. The response time here, we're using the intracellular calcium concentration response as indicator or as outcome variable to measure how cell respond. So you see that there's a very good correlation between the planar distance from the beam tip and the response time, right? So which means the closer you will have the cell response immediately, and the further the cell is not responding immediately, but later on they will get a signal. But the problem is, where do they get a signal? Do they get a signal directly stimulated by the beam generated a fluid flow shear stress? In that case, it will be very low. If you look at when it's going further, the shear stress level is much lower. Or they simply get some signals from the surrounding cell, which are closer to the beam team, close to the tip of the beam. So these cells are directly stimulated. They will release certain factors, which will be convectively transported to these other cells, and then they will get excited. So we don't know that. I forgot to embed it, sorry. So there's so we published this, actually, we published this in, I believe it's also 2019 or even earlier this year. And there's a video actually on the journal's publication, on the journal's website. So if you're interested, you're welcome to take a look to see how the system works. So that really says that with this very new novel device, we can actually not only look at whether ultrasound can respond to flow, how they respond to flow, but we can also look at under this very in vivo-like environment where the force is not homogeneous, how cells behave and how cells communicate to each other to coordinate the response. Okay, so that will be the real end of my talk, I guess.
Thank you very much, Dr. Lydon. It was very interesting and engaging talk. I actually learned a lot, and just the fact that you work with microfluidic devices, I am now just starting to work with them for neurons. So it's a very, it was very interesting to listen about it, and your application of the microbiology of bone in the cancer is actually very promising. So we have a question here from the audience. If there are any challenges in the miniaturization involved in the fabrication of the mini chip?
You mean the magnetic chip project, right?
The microchip that you developed. I believe it's the first.
Oh, the first one. Yeah, definitely. Actually, even though I covered it within 1 or 2 slides, but fabricate this device, if I can go back, fabrication of the device is actually very tricky because first, when you have the side channel hydrogel fused side channels, it's actually very easily to allow the bubbles to trap here. And in microfluid field, we absolutely don't want any bubble trapping. And just for that part, my students, Christina May, and she put a tremendous effort on improving design to make sure there's no bubble, and also the formation of this artificial lumen or vessel-like lumen structure is really non-trivial, right? So we want it to be circular in terms of cross-section. People may ask why it has to be circular. Of course, from the morphology, we want to make it circular to be closer, look like a vessel, but that's not the only reason, because it has been shown that the size and the shape of the vasculature you form is actually pretty important, determine the endothelial cells metabolism and function. So if you put them in like a square channel, even though they can still form this endothelium-like structure, but they don't totally do what they're supposed to do. So they actually, the sphere, the cross-sectional, circular cross-section is actually quite critical for the appropriate endothelial cell function. But in order to achieve that, so we have to do a lot of tricks to make that happen, and even involve some like a gradually turn to make sure the uniform distribution of the gel and also of the endothelial cells sitting on the gel to form this very nicely circular-like structure. Circular-like structure. Yeah.
Okay, thank you very much.
Sure.
I believe, yes, our participant is saying thank you. Is there any other questions that you would like to ask? Please just unmute yourself and ask. It's also fine. No, they don't have any questions. Okay, then thank you very much, Dr. Leiden, once again for your presentation. It was very interesting, and I believe that we could meet one day, not through Zoom, but in person.
Yeah,
that would be very nice for a conference. And I would thank as well the Clinician Engineer Hub for this organization, and also to the EIT Health alumni for providing with the Zoom, and for my colleagues who also helped me organizing this event. And I would like to say goodbye and see you one day again.
Okay,
Thank you very much
for having me.
Have a nice evening.
Bye.
Bye-bye.
Bye.